Again, good morning, all. My name is Shankar Shanmugam. I'm an assistant research professor here at the Mississippi State University. The title of my talk today would be Insect Frass and Frass-associated Microbiome, the potential use as biofertilizer and biostimulants in crop production. Just for your information, this is going to be a deep dive into project 15, which was already a funded project that I'm working on. Okay, there are various plant beneficial soil microbes that are commonly applied in agriculture and are often considered to be a promising alternative to fertilizers and agrochemicals. While the potential use of these beneficial microbes for sustainable agriculture is great, often the outcomes of the microbial application in the field is very inconsistent. As the industry production of insects for food and feed is increasing rapidly, it's expected that large quantities of insect-derived products might enter the system, and they have a great potential to be used as a soil amendment. Frass is an important end product, like I discussed before, in an insect industry, and its use as a biofertilizer has a huge share in the alternative fertilizer market. I have here listed some important characteristics of Frass that could be beneficial when used as a biofertilizer. It helps in nutrient cycling. There are some microbes that help in nitrogen fixation in specific crops. But the important, based on my exploration, the important characteristics would be the phosphorus solubilization in the soil. Studies have shown that Frass might contain microbial species that have the ability to solubilize phosphorus and make it more accessible to the plants. As you all know, phosphorus is an immobile nutrient, and microbial activity in Frass might enhance its availability in the soil. Another important property could be its use as a potential plant growth promoting substance, PGPS. Frass microbiomes may produce plant growth promoting substances such as phytohormones, enzymes that stimulate the plant growth and enhance plant resilience to stress. And overall, the Frass could be used as a soil conditioner. It can help improve soil structure by enhancing water retention capability of the soil, aeration, nutrient uptake by plant roots, etc. So it has a great potential in this biofertilizer market. So biofertilizer is a new market, and the fertilizer potential of Frass thus needs more research before a major upscaling could take place in the industry. So to meet these goals, the objective of this project was to characterize the Frass microbiome as affected by the source and industry treatments, and to study how the Frass quality affects the plant growth response, soil nitrogen availability, soil microbial health indicator, changes in the soil, and to characterize the biofertilizer potential of Frass by monitoring the soil biological activity and crop growth. So in this study, we actually proposed to study the potential effect of adding the insect-derived products to how it might affect the plant and how it affects its plant growth, how it affects the beneficial soil microbiome that's already present in the soil, how it might help in plant growth promoting activities, if there are any microbial antagonistic agents that are present in this Frass that might help in the alleviating soil-borne plant pathogens and insects, and how it's going to help the overall plant growth and reproduction. So these are all the the factors that we want to test and the methods that we want to use to study these factors. So this is how we planned this research project. Frass from different industry treatments and feedstocks diet were characterized for its microbiome diversity, functional diversity, and plant growth promoting characteristics. So this is actually the first year. The May would be the end of first year of this project. So we already finished characterizing the Frass. So for the second year, the promising combinations from the first year study will be used for a greenhouse trial to study the plant growth characteristics, nutrient supply dynamics, and specific soil functions that this amendment could provide that could be used as a biofertilizer. So here is a list of treatments. We received Frass from different insect sources, drying treatments, and diet sources. Thank you to the IAB members for providing the Frass samples. We have I think we had a good list of different treatments and diet sources that we could compare to study the microbiome diversity differences in these Frass samples. Okay. So I'll be showing some results from the first year study. To start with, first we characterize the Frass using biolog ecoplates. So this technique has this technique actually monitors the change in the microbial community functional diversity. It uses a 96 well plate, which has like 31 different carbon assays in triplicates. So we extract the Frass and add it to the each wells. The microbes from the Frass will utilize the carbon sources, different carbon sources in these wells. The microbes respire, and as a result, the tetrasodium reported dye that is present in these wells is reduced to form visible purple color. And this purple color development could be quantified using a spectrophotometer. So this technique actually gives a snapshot of functional diversity that depend on the carbon utilization pattern of microbes present in the Frass samples. It's an usual technique especially for high carbon feedstocks like Frass. So what we did is we inoculated each of these plates with the Frass extract, different Frass extracts, and then we incubated them over 120 hours and periodically took the spectrophotometer reading. And based on the average well color development, we calculate different diversity indices. So this is the result of the biolog study that we did. So we had insect species, different treatments like raw or air-dried Frass, and different diet sources. So both the insect species and diet sources had a significant effect on the Simpson Diversity Index based on the biolog ecoplate study. So from here, I'll be showing our DNA sequence results from targeted metagenomic analysis. We extracted the total community DNA from these Frass samples, and then we sequenced them using Illumina MySeq, and then we did the data analysis. So these are the results from the DNA sequencing. So this plot here shows the phylum level abundance across all the Frass samples. So as you can see, the Firmicutes and Bacteroidetes phylum were the dominant phylum across all Frass samples, which is typical of gut microbiome diversity that you can see. Me working with soil samples, we won't see this trend. We usually find a lot of proteobacteria and acidobacteria. Frass from black soldier fly samples had the highest Firmicutes abundance compared to all the other samples. Okay, here is the alpha diversity change of the bacterial communities across all the Frass microbiome, across all the Frass samples. So as you could see, Frass from the cricket samples had a significantly higher alpha diversity matrix compared to all the other samples. This could be due to the more amount of chitin present in these samples. We are still yet to characterize the chitin and the other compounds, but that's what we now anticipate to see. Okay, so here is an ordination plot showing the bacterial beta diversity. So these plots in an ordination graph, the points that are closer to each other, they represent microbial communities that are similar in sequence composition, and if they are farther apart, then the composition is different. So that's how we need to infer from these plots. As you could see, the cricket and BSF were forming a distinct structure. They were significantly different in their bacterial community composition. Okay, so this is the result from the analysis of the fungi. We also did the ITS sequence, ITS gene sequencing to understand how the fungal community might change across these Frass samples. Here, the mealworm sample, Frass samples, got a significant higher fungal community diversity compared to all the other samples. Okay, so this is the this plot shows the fungal beta diversity across all the Frass samples. BSF again showed a significant different clustering pattern compared to the cricket and the mealworm samples, showing that the community diversity or the community composition are significantly different between the BSF and cricket and the mealworm samples. Okay, we characterize the plant nutrient content of these Frass samples, and here is a table that shows the analysis. Along the rows, you have the different insect groups, and the column has the analysis of both the macro and micronutrients. As you can see, the Frass is not a significant source of the macronutrients like nitrogen, phosphorus, and potassium. This is like any other organic amendments. You won't expect very high levels of the macronutrients. However, unlike other organic amendments that I have worked with, I see that the Frass samples, they are very rich source of micronutrients. There is an increasing interest from the agricultural community in the micronutrient fertilization. There are various reasons for this. The soil erosion, long-term cropping have resulted in the removal of the micronutrients from the soil. Also, the increased crop yield generally, it removes the grains, and the harvesting material will remove most of the micronutrients from the soil. It's often difficult to replace that with straight fertilizers. So the farmers are being recommended with micronutrient fertilizers, which are very costly. So the micronutrients actually they are essential plant nutrients. Although they are found in very trace amounts in the tissue, but they play an important role in plant growth and development. So as an amendment, the Frass, it could be an important source of micronutrients. So I think this is very important property of the Frass. So we went ahead and did some general functional potential, which groups these microbial organisms to different functional regimes like carbon metabolizers and organisms that produce enzyme that are involved in carbon cycling, nitrogen cycling, etc. These are important essential characteristics of any organic amendments. So we tested the carbon cycling potential of these Frass samples, and we used PyCrust-2 as software for predicting the functional abundance based on the gene sequence from 16S database. So we found the cricket Frass samples had a significantly higher carbon cycling potential. This was very consistent with our biolog studies where we had a higher diversity of the cricket samples. Okay, here is another important characteristics that we studied. We looked at the potential N-cycling organisms in these Frass samples. Nitrogen is often, as you all know, nitrogen is often a limited resource for plant due to low soil nitrogen levels. So this would be an important characteristics of Frass when it is applied as a soil amendment. As you could see, the black shoulder fly, obviously had a higher, in metabolizers because the table that I showed previously, the BSF samples had higher N content than other Frass sources. So we would expect more of the N cycling organisms present in those soil, in those Frass samples. Okay, so there was a concern from the IAB members during my previous meetings that would there be any pathogens that might be introduced when we are bringing this as a soil amendment into crop production system or vegetable production system. And those concerns are really valid. So we went ahead and did an analysis where we could say if these organisms could be a potential plant pathogen. So we looked at especially the fungal community for their plant pathogen activity. So we analyzed the Frass and we put this database into a fung guild, which is it actually separates the sequences into different guilds of different functional groups, and the result here shows one of the functional groups, which is plant pathogens. There are different functional groups like soil-borne plant pathogen, airborne plant pathogen, etc. We looked at as a whole as pathotrophs. There are saprotrophs and everything, but we are just interested in pathotrophs here in these Frass samples. So we did find significant numbers of Bloomeria graminis in these Frass samples. As you could see that in all the Frass samples. This is a fungal pathogen that causes powdery mildew disease in grasses, in most of the grasses, I think. So we were wondering, where is this coming from? So we looked at the different diet sources, and we found, we were actually curious to see, where they're coming from. We looked at the diet source, and interestingly, actually, the Frass from insects that were fed with corn soybean diet showed the highest number of this particular pathogen. Well, however, these are low abundance numbers, I mean, the overall fungal population, as we are only showing the fungal guilds that would code for the pathotrophs. And these pathogens, they're not soil-borne pathogen, but it will be interesting to see their fate, when we are applying it as a soil amendment, and what happens to them when they're applied, when they start interacting with the native microbial communities. Will it cause eventually, if the pathogen load will be enough to cause plant disease, etc., that needs to be studied, and that's what we are going to study during our second year study, which is the greenhouse study. Okay, in a similar context, we did a core microbiome analysis on all the Frass bacterial data set, and we did find some human pathogens, but at very low prevalence level. If you look at this heat map, their prevalence level was close to like 5% or something. But we did find some Staphylococcus species in some samples, so we are still teasing out, whether we could factor it out to the direct source or any other treatments or the insects. We are still figuring it out. Here again, it has to be seen what would be their fate in soil. Studies have shown that when, especially this is true for using biofertilizers in agriculture. I mean, we usually see a rapid decline in the inoculant population activity following the introduction into the soil. It all depends on the rhizosphere dynamics. It'd be interesting to see how they are going to interact with the native microbial communities, and also whether it would eventually enter into the food system. So we'll be studying all that in our second year greenhouse study. Okay, during when we started characterizing the Frass, we got Frass from different industries. So these were having different physical properties. They had different texture, different aggregate stability, bulk density. The results that I showed above are from these Frass samples with different texture, moisture, regime, and bulk density. During our second year study, the greenhouse study, we will test to see if these characteristics will have any significant effect on the plant nutrient availability. So we'll also be looking at the physical properties of this Frass in terms of how they could affect the plant nutrient relation characteristics. So that will also be studied along with the microbiome change that it would incur when added as a soil amendment. Okay, so this figure shows our proposed greenhouse study for the year 2. So we'll have experimental blocks with different Frass soil combinations. So based on the nitrogen analysis of each Frass, we will apply different rates of Frass to the potted soil. We'll start from low rate to high rate, depending on the crop nutrient requirement. As of now, we are planning to study a fertilizer responsive corn crop, but we would also be interested in studying some a cold vegetable like a spinach, which is often used for biofertilizer studies. So we are also considering studying that crop. So this will be the experimental setup, and during the growth phase, germination will be recorded, scored, and the temporal soil sampling will be taken to assess the nutrient supply characteristics and the microbial characteristic changes over the crop growth period. And finally, we will be doing a lot of plant measurements, which will include leaf number, plant height, chlorophyll content. Fresh and dry biomass will also be recorded. And we'll be looking at how the rhizosphere microbial communities will change over the growing period. So we'll be extracting the rhizosphere soil. We'll be doing the DNA isolation. We'll be sequencing it to assess the microbial diversity across these different treatments. So that will be our greenhouse study. So we have actually started prepping for the greenhouse study. We have laid out the pots, and then we have set it to equilibrate. We will also be autoclaving a set of soil samples. So this is all in preparation based on the number of pots that will be able to work. We'll be looking at both natural soil, some potting medium, and also an autoclaved soil. So we have already started working on those. So just some final conclusions, and that would align with the future work on this project as well. Our results showed that the composition of the frass was strongly dependent on the insect species and feed substrate. Also, based on the frass microbiome assessment, we hypothesized that the application of frass would differentially affect the microbiome of the soil, both the soil and the plant rhizosphere interface. So these evaluations should not only be, we are doing a controlled experiment for the second year, but eventually it would be good to study them in a natural agro ecosystem in the outdoors, where the environmental conditions are variable. It's that way we can also compare it with the real life NPK, regular NPK fertilizer, and we are also hoping to extract and maybe develop a biostimulant model by extracting the rhizosphere and see which communities are favoring the plant growth parameters, and we'll be isolating those communities, and we'll probably be looking at if that could be developed as a biostimulant, that could be marketed as a biostimulant. So this frass has both the biostimulant and the biofertilizer potential, and that's what we think and we see based on our initial characterization, and that would be the path that we wanted to take in this project for future as well. So as we speak, the biofertilizers already they are in the market. They hit the market in Canada. Thanks, Michael, for sharing this link. So I like the name of the their fast. It's called Kicking Fast. So looking at the label, the nutrient analysis is actually very close to what we found in our frass samples. So there's a market for this. So on that note, I would like to acknowledge Dr. Dylan, who is a co-PA on this project, who helps with the soil fertility aspect of this soil and plant aspect of the study. And Jasmine Sahota, the graduate student working on this project. We would like to thank the IAB for funding this project and CEIF leadership for including us in this research effort.